Hello, kids. How are you? Here I am again with another new topic from grade 12 biology, unit 1, biotechnology, of course. Last time I have uploaded one video on tissue culture, of course, tissue culture, specifically plant tissue culture, that focuses on plant tissue culture, PTC. Now, next to tissue culture, health and well-being, human health and well-being, the next section, grade 12 biology. And therefore, we shall talk about this. When biotechnology used in human health and well-being, it is involved in two ways, of course, as you know, in the production of materials, production biotechnology, we call this, or the production biotechnologies, and the other is provision of service, to provide service to humanity, to make human life better and solve human problems. That is the service industry or the service biotechnology. Now, we focus, of course, on the first, in the production material, the production of materials. The production biotechnologies include production of some, in terms of health, of course, production biotechnologies, as far as human health and well-being is concerned, is production of some bioactive agents like human hormones, some drugs of animals and humans, antibiotics, animal hormones, and some other bioactive substances or chemicals produced in the biotechnologies as products to serve humanity in health and well-being. And human hormones, let us say some examples. Human hormone, as far as hormone is concerned, the immediate hormone that comes to our mind is insulin, which is produced by genetically engineering the E. coli being produced now, genetically engineering E. coli. We insert human insulin gene into the E. coli plasmid, the vector, and so this E. coli will reproduces faster in industries with the provision of relatively cheap nutrient source, so that it can produce huge amount of insulin for diabetes mellitus patients. And the insulin that is produced by genetically engineering bacteria, E. coli, has been named humulin, to identify it from the natural insulin, a protein hormone, of course. The other is human growth hormone. This is also protein hormone, human growth hormone. This hormone, human growth hormone, is produced naturally by the pituitary gland, which is a gland, sometimes known as master gland, found in the brain, below hypothalamus, of course, and pituitary gland, and promotes body growth, generally skeletal system growth, and also other body growth and development. That's why we call it growth hormone. It's used to treat body growth. We produce this human growth hormone to treat body growth retardation caused by deficiency in the production of the hormone. That is, in some individuals, children, because of hyposecretion of the pituitary, the anterior pituitary gland, hyposecretion of growth hormone, children may become very dwarf, short size, very dwarf, so that people don't like dwarfism. Of course, dwarfism is considered as one problem in our community. Therefore, if they want to treat, if they don't want their kids to be dwarf, it can be given hormone therapy, growth hormone therapy, and bring his or her average height to an average at least. But therefore, it is used to treat pituitary dwarfism, to differentiate from genetic dwarfism. There are other forms of dwarfisms transmitted because of errors in genes. This one is because of lack of sufficient production of growth hormone by pituitary gland. The other is tissue plasminogen activator. This is a protein. This protein is a crucial protein that helps dissolve blood clots and used to treat clotting related problems. Imagine, wherever there is clotting forming in the circulatory system, it's a huge problem. It brings about the hypertension, the silent killer, and also stroke, heart attack. In order to prevent all these deadly cardiovascular problems. Whenever there is clotting, we can treat clotting by injecting this into the circulatory system, tissue plasminogen activator. The other is in area of animals, animal hormones, bovine somatotropin or bovine growth hormone. This is growth hormone produced by pituitary gland of cattle and controls body growth, muscle mass increase, and also in cows milk production is enhanced by this bovine somatotropin. So in agriculture, what does it mean? In agriculture, increasing flesh production or meat production plus milk production means one solution in terms of nutrition for humanity. Agriculture productivity increase. The other is porcine somatotropin. The term porcine refers to the pig growth hormone. In order to increase the size of pigs, especially in order to increase their muscle mass, we use the growth hormone produced by pituitary gland. By first, we produce the pituitary gland. We produce this hormone, porcine somatotropin, or the other hormones are produced by genetic engineering at industrial scale by genetically engineering bacteria. Then the product is injected into cattle in order to increase productivity, into pigs in order to increase PST, that is, porcine somatotropin, plays a crucial role in regulating growth and metabolism in pigs, leading to increased muscle mass and reduced fat deposition, of course. It is being produced by genetically modified bacteria so as to be used to boost meat productivity in pigs. Some drugs, some other bioactive agents, some drugs and antibiotic production. Cyclosporin is another drug used by humanity as immunosuppressant. Cyclosporin A, they call it, is an important bioactive chemical used as an immunosuppressant agent in organ transplant. You see, there is one problem in organ transplant. When we are transplanting organs like kidney from one donor to the recipient's body, the immune system, reacts in a negative way to reject, which is known as immune rejection. One big problem of organ transplantation is immune rejection in medicine. Then, how can we solve the immune rejection? How can we be sure the body of the recipient, the immune system of the recipient, will not produce antibodies and destroy the transplanted organ? We give continuously, the person who receives the transplant organ receives immunosuppressant drugs. Therefore, one of the immunosuppressant drugs, that means drugs which decrease activity of the immune system, is cyclosporin A. This is the solution, immunosuppressant drugs. The cyclosporin A, its production, when you see cyclosporin A has been synthesized in large quantities by fermentation process using various fungal species like Tolipocladium, Trichoderma, Fusarium, Penicillium, and also Aspergillus. Imagine Penicillium, Aspergillus, these are the two genera very well known in meat fermentation, penicillium in antibiotic production, also Aspergillus. Therefore, these are really diverse. Therefore, these are the common fungal species that are used industrially in the production of cyclosporin A, which is bioactive agent that suppresses the immune system in case of organ transplant to stop transplant rejection. The other bioactive chemical is statins. Statins, this is of course a class or a family of medications, of course. Statins are a class of medications that effectively lower cholesterol level, especially in obese individuals where the bloodstream contains huge amount of cholesterol buildup, which is not good. Cholesterol increase in the circulatory system means high blood pressure, cardiovascular problems, including stroke and heart attack. These statins are competitive inhibitors of the key enzyme in human body that is involved in cholesterol synthesis. If you can inhibit the key enzyme HMG-CoA, which is known as they CoA reductase, the key enzyme in cholesterol synthesis. If you inhibit, therefore you inhibit the production of cholesterol in the body. That means cholesterol will not rise or increase in the body and cause cardiovascular problems. Therefore, statins are many different types. Of course, statin is nowadays produced synthetically, but the original source of this chemical are these two fungi still, Aspergillus and penicillin. Imagine, they are miracle fungi. While many statins are now produced synthetically, their natural precursors were predominantly produced due to fermentation by Aspergillus and penicillium fungi species. But now, the drugs are produced synthetically in the lab. Example, natural. Mevastatin is one type of statin from the fungus Penicillium citrinum. Lovastatin is one type of statin from the fungus Aspergillus terreus. You see, this is two typical examples. There are many others. Now we shall go to antibiotic production. The production process, antibiotic production. What are antibiotics? First, let's first define. The term antibiotics is derived from or built from anti and bioti or biotas. Anti means against. Bioti or biotis means life. Biotic, biotica, biot, life. Therefore, literally, the term antibiotic means literally against life. Of course, this is a wrongly given terminology or name, wrongly given. Antibiotics are not totally against all forms of life. Antibiotics are primarily inhibiting or killing what? Bacteria. We are using antibiotic to counter, to be against bacteria, pathogenic bacteria. Of course, we are using them, disease-causing bacteria, to counter. However, it says this is a misnomer, as antibiotics are chemical substances that are against bacteria, not against all forms of life. Therefore, wrongly given name. Antibiotics are chemical substances or metabolic products secreted by some microorganisms. Some microorganisms. What are these some microorganisms? If you ask me, of course, it includes many different types of bacteria, including bacilli, and also some molds, a group of fungi known as single-cell fungi known as molds. For example, penicillium is a mold. And also special types of bacteria which do have fungal characters known as actinomycetes, filamentous bacteria which do have some sort of character of fungal actinomycetes. These are the three major groups of microorganisms that are employed in biotechnology for production of antibiotics. Then antibiotics are produced and counter bacteria. How do they counter? They either inhibit the growth of bacteria. If an antibiotic inhibits the growth of bacteria without killing it, inhibits the growth or multiplication, it is known as bacteriostatic. So that the number of bacteria will not increase and cause serious problem. Inhibit the growth of bacteria. Such antibiotics are known as bacteriostatic. The second group directly interfere and kill bacteria in many different ways. These are known as bacteriocidal. Cidal means killer. They kill. No mercy. No mercy. Which microorganisms are known to produce antibiotics? I already told you bacteria. Many different groups of bacteria are there here, including bacilli, fungi or molds, such as penicillium, and actinomycetes, which are filamentous bacteria that resemble microscopic mold. Antibiotics are not fermentation products. Be careful. They are not fermentation products. They are fermentation products are primary products, primary metabolites. Metabolites refers to products of a chemical reaction. Primary metabolites are those fermentation products which are essential for growth, reproduction, energy production, whatever. They are essential. Microorganisms can survive without primary metabolism without primary metabolites. Antibiotics are secondary metabolites. That means they are not that much essential for growth and reproduction, but produced and secreted and released out for various purposes. We shall talk about that. I'm telling you, generally secondary metabolites. Antibiotics are not the only types of secondary metabolites. There are many different types of secondary metabolites almost in all groups of organisms, in animals, in plants, in fungi, microorganisms, of course. Therefore, antibiotics belong to the group of metabolites known as secondary metabolites. They are not essential for survival, just they produce, microorganisms produce secondary metabolites like antibiotics. They produce antibiotics as secondary metabolites for what purpose? Of course, no clear-cut answer. There are some theories with not much substantial evidence, of course. Secondary I've told you, secondary, first you have to be clear about what secondary metabolites are. Secondary metabolites are organic compounds synthesized by bacteria or other microorganisms or even plants, animals. And unlike primary metabolites, which are essential for growth and reproduction, secondary metabolites are not directly involved in these processes. That means in growth and reproduction, but serve various ecological functions. You see, ecology, underline that term, ecological functions. For example, some secondary metabolites are used for communication among microorganisms around the area. Bacteria may produce some chemicals to talk to each other, to interact. Some chemicals are secondary metabolites. For example, there is a process known as quorum sensing. Bacteria produce and release out chemicals for quorum sensing. What is that quorum sensing? They are releasing these chemicals to assess the situation in the environment. To sense the environment, the population density, whether there are nutrients, what sort of variety of bacteria or microorganisms are there around them. Therefore, this is very important, ecological surface, ecological function. And when it comes to antibiotics, which kill bacteria, why do bacteria, molds, and actinomycetes produce antibiotic? Not clearly known, but there is one theory, as I have told you. Why do microbes produce the antibiotic chemicals? The reason is not clearly known. However, one theory is that the microbes produce the antibiotic chemical to negate, to oppose others, so that they can reduce competition for nutrients in the area and space. Just to compete and win for the nutrient and the space around. Types of antibiotics. Again, one category, one way of grouping antibiotics is based on spectrum of activity. How much spectrum of activity means how much variety or different groups of bacteria are killed by specific antibiotic. Is the antibiotic only killing a small number, only one type, just two types? No. Can it kill many different types? That is broad spectrum. We prefer broad spectrum, of course, but it has its own negative consequence. Broad spectrum antibiotics, when we take them, they also kill our microbial friends inside us. You see, the human body, the GI tract, small intestine, especially the large intestine, is full of bacteria. You have 10 bacteria for every single cell in your body. 10 bacterial friends. And these most are not harmful. Even some are useful. Very important for you. Therefore, when you take broad spectrum antibiotic, you may also kill your microbial friends. You see, so that you decrease your microbiota. That means the normally living microorganisms inside your body, inside the GI tract, of course. Therefore, anyway, let's go. This classification, antibiotic classification 1, based on spectrum of activity, narrow spectrum antibiotics. They are effective only against specific types of bacteria. For example, penicillin G. Only targets or inhibits or kills gram-positive bacteria. There is gram reaction. We will come to this point: gram-positive, gram-negative. We will come to this staining technique. Grams staining technique divides bacteria into gram-positive and gram-negative. Therefore, this only is active against gram-positive bacteria. Because of their cell wall chemistry. Penicillin works by inhibiting cell wall synthesis. Therefore, it can perfectly inhibit cell wall synthesis in these gram-positive bacteria. The other broad-spectrum antibiotic effective against a wide range of bacteria, which includes gram-positive as well as gram-negative. Tetracycline is one type of broad-spectrum antibiotic. How do antibiotic drugs work? The mechanism of action. How do antibiotic drugs either inhibit the growth, bacteriostatic, or kill the bacteria, bactericidal? How do they do that? The mechanism of action of antibiotic drugs at molecular level. One is by inhibiting cell wall synthesis. Some drugs, for example, described here, vancomycin, cephalosporin, monobactam, and penicillin. These drugs kill bacteria by inhibiting the enzymes that make the cell wall of bacteria. They are killers. If bacteria have no cell wall, water enters by osmosis into the cell of bacteria, builds pressure, and the cell membrane it can't resist pressure. It breaks, so that lysis, bacteria will be killed. These are killers. These are sida, bactericida, vancomycin, cephalosporin, monobactam, and penicillin. How do they kill? By inhibiting enzymes that work on the cell wall synthesis, that work cell wall of the bacteria. If the enzymes are inhibited, the bacteria will not have cell wall, water enters, and it bursts. Osmotic burst of the bacteria is killed. The second group, by interfering with protein synthesis. Some antibiotic drugs interfere in the bacterial protein synthesis by attaching on bacterial ribosomes or bacterial enzymes that work on protein synthesis. They inhibit synthesis of proteins of bacteria. From this category, chloramphenicol, tetracycline, streptomycin, aminoglycosides, macrolides, these are the protein synthesis inhibitors. You have to know at least this. The third group, as example, by interfering with nucleic acids, and the nucleic acids involves both DNA and RNA. As some inhibit DNA synthesis, the others inhibit RNA synthesis or DNA transcription inhibitors. There are. Anyway, they inhibit nucleic acid synthesis. In this group, examples are quinolones, rifamycins, sulfonamides. There are many others, of course. This much is enough for you as an example. Now, we shall go to the last part, very brief history of penicillin. Before we talk about the production process of antibiotic, very brief history of penicillin. Penicillin is the first antibiotic drug accidentally discovered in 1928 by Alexander Fleming. Therefore, the history of pharmacology begins from here. Thank you very much. I will come to the portion next to this later on. For today, this much is enough. Thank you very much. Please stay awake, work hard, and if you do this, there will be plenty to eat. Stay awake, work hard. There will be plenty to eat. Thank you.